Hey everyone, so this is a test run to see if I can record simple lessons using the whiteboard app on my brand new fancy tablet. I wanted to go over the genetics lectures with you just briefly to make sure that you picked up on all the really important points and to show you what you're going to need to be able to do in order to pass the exam. So what the lectures focused on at first was what we call Mendelian genetics. And Mendelian genetics involves the interaction between two different types of traits, dominant traits and recessive traits. Now, before we can even really talk about these, we have to discuss those two, or that word that I was just using, which is trait. There are two ways to describe the physical attributes of a living thing. You can talk about their characters, which are sort of like categories. So for example, if I were to say the words eye color to you, you'd be able to picture the structure I was talking about, right? An eyeball. And you would know that I was talking about the color of the iris in that eyeball, but you wouldn't be able to tell me what color I was discussing because I didn't name one. Eye color is a category that includes different color possibilities. You can have, you can have blue eyes. Here, I'll just stay with this color pen. You can have brown eyes. You can have green eyes, so on and so forth. You've got options here. Eye color is the character, it's the category. But these specific versions of eye color, these are what we call traits. Traits are these specific versions of a character. So the term character is more general, the term trait is more specific. Now, going back up to our first point up here, dominant and recessive, these are terms that refer to traits. You can have a dominant trait, which, when it's present, is the one that controls the physical appearance of that particular character. You also have recessive traits. When a recessive trait is present, it's usually hidden if you also have the dominant version of that trait. Now, how are you going to wind up with both, a dominant and a recessive version of a trait? Well, the reason this happens is because humans and most other animals are what are called diploid organisms. If you're diploid, it means that you have two copies of your DNA. Now, some organisms don't have this. Some organisms only have one copy of their DNA. Those organisms are called haploid. Okay. So here's how this works. Your DNA inside of the nucleus of your cells, in fact, let me color code this. Okay. So this big circle, this is the nucleus. There's this really common misconception that DNA is one big, long piece of one giant chain of nucleic acids that just goes and goes and goes. This is not correct. It's a really common depiction of your DNA in cartoons and in animations, but it's not accurate. Okay? Instead, your DNA is divided into pieces. Humans have 23 different pieces of DNA, but not all organisms do. Some have 36, some have 12, some have closer to 50 individual pieces. But we'll use humans as an example. Humans have 23. And those 23 pieces are all different sizes. Okay? And each piece we can distinguish from every other piece based on its size. Now, these pieces of DNA don't float around alone in the nucleus. Instead, the DNA is wrapped around a series of protein balls. So these little guys are all proteins. And the DNA itself loops around them like this. This is to keep the DNA organized, to keep it from breaking or tangling up. And it gives you, once it's completely wrapped around these proteins, a nice tight little bundle that winds up almost looking like a piece of yarn wrapped around some sort of central structure, like a chopstick or a piece of wood. But it's a nice tightly bound bundle. And it's a mix of just this DNA and these protein balls in the middle. Okay. Now, we have a name for this material. Whenever you add these protein balls to this DNA, what you've made is a new material that we call chromatin. Okay, so that's the actual material. This structure, though, that we've just created, this bundle of DNA wrapped around these proteins, that has a name too. We call that a chromosome. And since your DNA hangs out in your nucleus in this form, when we talk about all these different pieces of DNA, we don't talk about there being 23 pieces of DNA in your nucleus or 23 different pieces. We talk about there being 23 different chromosomes, since the way the DNA occurs naturally in the nucleus is in the form of a chromosome. Okay? So, humans, go back to my original color. All right. So humans have 23 different chromosomes. We can arrange them by size. Okay. I'm just going to draw out a few for you. So let's call this chromosome number 1. The next one, number 2, and number 3, so on and so forth, until you get to the last two, numbers 22 and 23. Okay. So each of these chromosomes is a different size, but more importantly, each of these chromosomes has different information on it. For example, remember that these are chunks of DNA wrapped around proteins, and that DNA contains information. This spot on this chromosome could be the location in your DNA that codes for that eye color we were talking about. If this is the section of your DNA that codes for eye color, we would call this your eye color gene. Remember back when we were talking about macromolecules and we talked about nucleic acids? Genes are sections of your DNA that contain the information for building particular parts of your body. Now, the eye color gene is on this location of chromosome number 1. Now, what if we went to a similar location on chromosome number 2? Would we find another eye color gene there? No. We'd actually find something totally different. This location on this chromosome is going to have a completely different gene on it than that same location on chromosome number 1. This might be the gene for height, whether you're short or tall. Likewise, if you go on to the next chromosome and go towards the top of that chromosome, you'll probably find, or you will definitely find, a completely different gene. Maybe this is the gene for shoe size. Okay. Each of these chromosomes is going to have different genes on it. No two chromosomes will have the same genes. Okay? So you've got 23 pieces of DNA that are all different from each other. But remember, I said humans were diploid organisms, right? And I said that when you're diploid, you've got 2 copies of your DNA. So what does that mean? It means that for humans, being diploid translates to us having 2 copies of every chromosome. So we've got 2 copies of chromosome 1. We've got 2 copies of chromosome 2, 2 copies of chromosome 3, so on and so forth. Okay. Both copies are going to have the same genes on them. So chromosome 1 we set up here has the gene for eye color. That means on this spot, on this chromosome, this will still be the gene for eye color. Oops. And over here, on this copy of chromosome 1, we're also going to have a gene for eye color. We said on chromosome 2, in that similar location, right here, that was the gene for height. On this copy of chromosome 2, it's also going to be the gene for height. Chromosome 3, here, shoe size. Same thing on this copy. That location will also have a shoe size gene. So what this means is that not only do we have 2 copies of all of our DNA, that means we have 2 copies of every gene. 2 copies of the genes that code for our eye color, 2 copies of the genes that code for our height, and for every other aspect of our physical body. Okay, here's how this relates back to where we started, up here, with dominant and recessive traits. Okay. Remember that genes, let's write this in a different color. Remember that genes are sections of your DNA that code for a specific character. Okay. So genes code for characters. But remember, characters are just categories. Specific versions of a character are called traits, and traits are coded for by different versions of the same gene. Because it's science, we have to give a special name to everything. We have a name that we give to those different versions of the gene. Those different versions are called alleles. So just to really spell this out, to make sure it's totally clear, I'm going to write it big. Traits are different versions of characters while alleles are different versions of genes. Oops. Now there's one more thing we've got to add to that, but I'm going to add it in a different, no, I'll keep it in the same color. All right, one more thing. Connect it to this box. It's how these concepts connect to each other. Genes are the regions of your DNA that code for characters. So we'll just say genes code for characters. I know I wrote this above, but it's important. I'll write it again. Now, if genes code for characters, and you know that characters have different versions called traits, hopefully that leads you to conclude that the different versions of a gene, alleles, are what code for those traits. Genes code for the characters, different versions of those genes, alleles, code for the different versions of those characters, which are called traits. And that's a really critical connection to make. This relationship between genes, alleles, characters, and traits. So it's in this orange box. Definitely make sure you know it. So here's an overview of what we've talked about so far. I want to go back up to our starting point up here and start relating these ideas of genes and alleles and characters and traits back to how characteristics are inherited by offspring from their parents. So let's come back down here where we had just started talking about what it meant to be diploid, that you have two different copies of every chromosome, and they have the same genes on them, but the trick is that you can have two different versions of the same gene on each chromosome. So what does that mean as far as how your DNA affects your physical appearance? Well, let's take, again, chromosome number 1 as our example, okay? And let's talk about eye color. So just to use the terminology that we have introduced, okay, our character is going to be eye color. And just to simplify things, let's say that there are two possible traits for eye color. Let's say the only possible traits are brown and green. Okay? You can either have brown eyes or you can have green eyes. Those are your two options. I'm going to redraw chromosome number 1 down here. Remember, this is copy number 1 of chromosome 1. Let's call it 1A. This is copy number 2 of chromosome 1. So it's still chromosome 1. But we'll call it 1B. Here's our eye color gene. Okay, so the same location on each chromosome. And that location, I'm going to throw in another term for you here. This location on the chromosome, where you find a specific gene, that's called a locus. Okay, so think location, locus. All right. So you know both of these sites code for eye color. The trick is, on this chromosome, it could be that this version of the gene, this allele, codes for brown eyes. But over here, this allele codes for green eyes. Now remember, both of these chromosomes are in your body. Both of these belong to you. So how does your body reconcile which color your eyes should be? In Mendelian genetics, go to purple. In Mendelian genetics, traits can be either dominant or recessive. If a trait is dominant, that means it controls your appearance. In our example, brown eyes are actually dominant over green, so brown would be our dominant trait. Recessive traits are masked or covered up or hidden by dominant traits. So in this example, green would be our recessive trait. Okay. However, you know there are people with green eyes. It exists. So how is that possible? Well, the only way that you can have a recessive trait control your physical appearance, so the only way to have a recessive trait appear is if you inherit from your parents two copies of it. And when I say it, what I mean is you inherit two copies of the recessive allele. Clean that up. Now, we use shorthand when we're talking about genes and representing dominant and recessive genes because we don't want to be writing out the words brown and green over and over again. And just by their names, we don't really indicate which one is dominant or recessive. So to try to make this more clear, to keep our language consistent, we are going to use an uppercase B to represent not only the brown-eyed allele, but also the fact that that allele is dominant. Capital letters represent dominant traits. Likewise, down here, the green allele, which is recessive, we're going to represent with a lowercase letter to represent that it's recessive. The trick is we also want to indicate that these letters belong to or refer to the same gene. So we're actually going to use the same letter. We'll use a lowercase b to represent green eyes, uppercase B to represent brown, and it's just understood that the letter B represents the character eye color, or the gene that controls the character for eye color. Now, how do you know if your offspring are going to have the dominant version of a trait or the recessive version? Well, there are ways to figure this out. Let's say you have a female, a mother, who has brown eyes. She's got to have at least one dominant copy of, or at least one copy of the dominant allele in order to have those brown eyes. But what's interesting is she could also have either a second copy of the dominant, meaning both copies give her brown eyes, or she could have a recessive version of the allele as her second copy and still have brown eyes. Because remember, the dominant allele is going to block the recessive allele and keep it from expressing itself. Okay? So let's use this as our example. Let's say the mother has both copies, or she has a copy of each, a copy of the dominant and a copy of the recessive. When you have one of each type of allele, dominant and recessive, we say that you are heterozygous for that particular gene. Hetero means different, okay? It's because you have two different alleles for that one gene. Now, the father, let's say he has green eyes. Now remember, there's only one way to get green eyes. If he has any copies of the dominant allele, he's going to have brown eyes automatically if this is a Mendelian trait. The only way for him to have green eyes is to have two copies of the recessive allele. Anytime you have two copies of your allele, two recessives, or if these were two dominants, we would say that you were a homozygote, or you were homozygous for that trait. Homo meaning same. You've got two copies of the same allele. Okay. All right, so here's your mother and your father. They're going to have children. If you want to predict what eye colors their offspring might have, you're going to use a little tool that we call a Punnett square. Now here's how you do it. Hopefully you remember from the meiosis lectures and videos that the alleles, or the two different versions of genes for a parent, separate when they form their gametes. So for example, this set of alleles is going to be separated into two different eggs that the mother's going to produce. One egg will receive the uppercase B, the other egg will receive the lowercase. The father, he's going to be making sperm, and his two alleles are also going to separate, one going into each of two different sperm cells. Okay? So all of the mother's eggs are going to look like this. They'll have either the dominant allele, one copy, or the recessive, one copy. The father's sperm are actually all going to be the same. They'll all have a copy of the recessive allele because that's all he has to give. Now, when fertilization occurs, an egg is fertilized by a sperm, and the two add together their genetic content. So let's say this egg happens to be an egg with a capital allele in it, or a dominant allele from the mother, and this sperm happens to have a lowercase allele from the father. When this fertilization event happens and these two join, the offspring is going to have a combination of the dominant and the recessive, one from mom, one from dad. And this will be their genetic composition for eye color. But obviously, this isn't the only possible combination. Remember, sperm and egg fertilization, it's random which of these eggs will wind up with which of these sperm. It could just as easily be a lowercase b, a recessive allele from mom, will fuse with a recessive allele from dad, giving you 2 recessives. So how do you sort out all these different possibilities? That's where the Punnett square comes in. You're going to take one parent and put their possible alleles on the top. You're going to take the other parent and put their possible alleles on the side. So let's put mom up top. One allele that she could pass on to her offspring is the dominant. The other allele she could pass on is the recessive. Now for dad, both of his alleles are recessive. That's all he's got to give. Okay. Now each of these squares represents a possible combination of alleles that an offspring could inherit. So for example, this top square represents an offspring that happens to inherit the dominant allele from mom and the recessive from dad. This square represents offspring that would inherit this dominant allele from mom and this recessive allele from dad. Over here, you've got the recessive from mom, and then go all the way over to the dad's side, you've got the recessive from dad. Okay, so here and here. Same thing, you've got the recessive from mom down here, and then the recessive from dad down here. These are all the possible genotypes. Okay. In fact, let's put that in color because that's important. Genotypes that your offspring can inherit. A genotype is the actual set of alleles that an individual has for a certain gene. Okay, they're actual types of genes. In this case, we have one offspring here who has one copy of the dominant allele, which is all it takes for you to have brown eyes. Over here, we've got no dominant alleles, just recessives, which gives us green eyes. Here, again, we've got that one copy of the dominant. That's all you need to get brown eyes. And here, again, 2 copies of the recessive, which gives you green. These traits that describe the actual physical appearance of the individual, we call that a phenotype. So the phenotype is the physical appearance. Think PH for pheno, PH for physical. This is what you actually see. Okay. Usually we represent the results of a Punnett square using ratios. The ratio of homozygous dominant individuals who have 2 copies of the dominant allele, and we compare how many there are of them to the number of heterozygotes who have 1 copy of each. And then we compare them to the number of homozygous recessive individuals who have 2 copies of the recessive allele. For this particular Punnett square, we don't have any homozygous dominant individuals. There are none. How many heterozygotes do we have? We've got 1 here, 1 here. That's 2. How many homozygous recessives? Well, we've got 1 here, 1 here. That's also 2. The overall ratio is 0 to 2 to 2, but you can simplify this. You don't have to write out the 0. If you leave it off, it's implied that there were no homozygous dominants. And instead, you can write this as either just 2 to 2, or you can simplify it by dividing each side by 2. Okay. 2 goes into 2 once. 2 goes into 2 again once. So your actual ratio here is for every heterozygote offspring, these parents are likely to have 1 homozygous recessive offspring. Okay. So this is what I expect you to be able to do when it comes to these basic Punnett squares. Okay. Let's do another one, just for practice sake. Okay. Let's create a separate space here. All right. So here's practice. What I'm going to do is give you two genotypes for a mother and a father. Okay, here's one. Here's the other. The X means you're going to cross them, meaning you're going to breed them. I'll draw out the box for you. I'd suggest pausing the video now and seeing if you can fill this box in on your own without me. Okay. All right, let's go ahead and do it. For this parent, we're going to assume this is the mom, we'll put her on top. Here's one of her alleles. Here's the other. This parent we'll put along the side. Here's one of his alleles. Here's the other. And then you just match the sides of the boxes. So the top of this box has the dominant, the side has another dominant. For this box, the top has the dominant, the side has a recessive. Over here, top has recessive, this side has the dominant. Now notice how I always write the dominant first? That's how you format these, okay? It's like putting a period at the end of the sentence or capitalizing the first word in a sentence. You just always write the dominant first. All right, down here, two recessives, one from above, one from the side. So what's our ratio? Well, homozygous dominant is the number that goes first. We've got one of those. Heterozygotes go second. We've got one, two of those. Homozygous recessive is our last one. We've got one of those. Okay. Now, this is your genotypic ratio. It's showing you how many of each genotype you have. However, you can also write out a phenotypic ratio. The phenotypic ratio is the number of brown-eyed children versus the number of green-eyed children. So you're looking at the actual phenotypes, the physical appearance of the individuals. Now, we have 3 possible genotypes, but only 2 phenotypes. And the reason is this pesky dominant allele. Wherever it's present, it controls the phenotype. So whether you have 2 copies of it, meaning you are homozygous dominant, that'll give you brown eyes. But even if you just have 1 copy, if you're a heterozygote, that will also give you brown eyes. So our 1 homozygous dominant and our 2 heterozygotes add together to all have brown eyes, meaning 3 of these offspring will be brown-eyed, and just our homozygous recessive individual will have green eyes. So make sure you keep these straight, the difference between genotypic and phenotypic ratios. All right. Now, what I've just shown you, all of this so far, has been what we call Mendelian genetics. And I know I've used that term before, but Mendelian genetics is when you have a clear dominant version of an allele and a clearly recessive version of an allele. But that's not always true. Sometimes you can have what we call alternative forms of dominance. This is where the dominant alleles don't always behave in a simple, straightforward fashion. For example, you can have what's called incomplete dominance. So what's this? All right, flowers are a great example to use when talking about forms of dominance. Right. So let's say you have a type of flower that can be either red or white. Okay. Now, if you were to cross these two flower types, okay, so you were to have them breed, something interesting would happen. If these are an example of incomplete dominance, if you were to breed these two flowers together, your offspring would not be red, and they would also not be white. Instead, they would be, if I can get my colors to pop up for me, there we go. There. Instead, your flowers would be pink. What the hell? So what just happened here? Well, think about it. What happens when you mix red with white? Well, if you're not using a tablet, the two colors would mix together and they would give you a shade of pink. Incomplete dominance is when your dominant alleles, in this case R for red and capital W for white, both of these are dominant alleles. There is no recessive here. They're both dominant. And instead of one covering up the other, they mix together. Neither is a winner, neither is a loser. They just blend into a phenotype that's halfway between the two original parents. Okay? This is incomplete dominance. Nobody gets to control the phenotype. Neither of these alleles are in charge. Instead, they have to share control. Now this gets interesting when you do a Punnett square for an incompletely dominant trait. So let's say you have two flowers that are pink. So two of these individuals, and you want to cross them. So we'll take one pink parent and we'll put their alleles up here. We'll take another pink parent, we'll put their alleles along the side. And let's see what we get. Well, this individual would be homozygous dominant red. They would definitely be a red flower. Let's skip to the bottom corner down here. This individual would have two copies of the white allele, homozygous dominant white. They would definitely have white flowers. But then our two individuals in the corners, they would be a mix. They'd have one of each. These would be our individuals who are pink. Now, if we write out a phenotypic, so in this case, we're just going to focus on phenotype. So if we write out our phenotypic ratio, it looks something like this. We have three phenotypes here. We have red, and how many red individuals do we have? Just the one. So we have one red flowering individual. Then we have two pink individuals. And we have one white individual. And this sometimes messes with people's minds, that you can have two parents. So remember, the parents here were both pink. but their offspring can be a mix of three very different colors. Okay. All right, there's one other form of alternative, one other alternative type of dominance I want you to know about. It's called codominance. If you can think of co as think of co as cooperating, maybe, or coexisting. Coexisting is better. In codominance, both of the original phenotypes will be present in the offspring. They're not changed in any way. Let's use flowers again. In fact, we'll use our same two types of flower. Our red flower, and then we're going to cross it with a white flower. Now, let's say in this situation, instead of being incompletely dominant, these flowers are now codominant. Here's how that's going to look different. We still have our dominant R red allele, we still have our dominant W white allele, but the offspring that will inherit, in fact, let me make this even clearer. This individual has 2 copies of the red allele. This individual has 2 copies of the white. Same down here. Everybody's got to have 2, right? They're all diploid. So the offspring receives 1 of each. But instead of being something that's halfway in between the phenotypes of the parents, this flower is going to be sort of a patchwork of the parents' phenotypes. There'll be blotches of red, there'll be blotches of white. The petals will be a mix of the 2 colors. Notice that the original parental phenotypes are still here. There's not a 3rd color that has shown up. The flower is still either red or white, depending on which part of it you're looking at. Okay? So there is shared dominance. Both original phenotypes are coexisting with each other, and they're both present. Okay, so that's codominance. The Punnett square for it winds up looking exactly like this one, only instead of having pink, your intermediate mixed color, you wind up having this patchwork pattern instead. Now, not all genes are carried on normal, what we call somatic chromosomes. Some of our genes are carried on our sex chromosomes. Those genes code for characteristics or traits that are linked to which of our sex chromosomes they're attached to. Remember, your sex chromosomes are either X or Y. Females have 2 X's. Males have an X and a Y. And this is really important because the X and the Y are different chromosomes. They're going to carry different genes, and that's really, really important to understand. There are some characteristics that are attached to only one or the other of these chromosomes. So you're only going to get 1 copy of that gene if you only get 1 copy of that chromosome. For example, Let's do flies. So fruit flies are these, I think, adorable little flies that can have either white eyes. This is my little fly head. They can have either white eyes or, same fly, can have red eyes. Funky looking flies. Okay. Now what's interesting is eye color for these flies is a gene, or is coded for by a gene on the X chromosome. Okay. If you have white eyes, you have 2 copies of the recessive version of this gene, or you have just 1 copy of it, and I'll get to that in a second. If you have red eyes, it means you've got at least 1 copy of the dominant, capital R, red allele. The white allele, the recessive, we're going to call that lowercase r. So in this case, capital R, dominant, will be red, lowercase r, recessive, will be white. Now check this out. Let's do a Punnett square for a male and female fly. Okay. The female will go up top. Actually, let's put the male up top. Female will go along the side. Notice I'm just writing out their sex chromosomes for now. Then I'm going to go back and fill in what version of the allele what allele they have for eye color on each of these chromosomes. So let's say for the female, let's say the female was the one with the red eyes. She's got to have at least 1 copy of the red allele. We write X big and the allele letter R small to indicate that this is a sex-linked trait. It's meaning the trait is literally linked to one of your sex chromosomes. All right, just for fun, let's say her other X has a lowercase r on it. She's heterozygous. Okay. Now the male, we already said he had white eyes. That means that if this is an allele carried on the X chromosome, his X chromosome must have the recessive version of the allele. Why? Because his Y chromosome doesn't carry that gene at all. In order for him to have eyes of any color, that colored allele has to be on his X chromosome. And if he's got white eyes, it tells us automatically that it's the recessive version, because this X is the only one he has. All right, so if we fill out this Punnett square, what we're going to get are half the offspring will be female, potentially. The other half will be male. Now let's see how the chromosomes or the alleles fall out. So we're going to have 1 dominant and 1 recessive here. We're going to have 2 recessives here. 1 dominant here and only 1, because again, the Y doesn't carry this allele. Down here, we'll have 1 recessive, and again, only 1. So what are the appearances, the phenotypes of these individuals? Well, this female, she's got at least 1 copy of the dominant, which means she's going to have red eyes. This female, on the other hand, she's got 2 copies of the recessive. She'll have white eyes. Now let's look at the males. They're where things get interesting. This male, he's only got 1 copy of the allele, but it's the dominant, so he'll have red eyes. But this male down here, he also only has 1 copy of the allele. It's the recessive. There's no dominant around to mask it, so it gets to control the phenotype. This male will have white eyes. So sex-linked traits are interesting for this reason. The males You can tell what allele they have for a certain gene if it's sex-linked based on their phenotype alone. That's not true for females unless they have 2 copies of the recessive and you can see the recessive trait. Okay, the last topic I want to go over with you is the dreaded dihybrid cross. This is a special kind of Punnett square, deals with normal Mendelian genetics, so normal dominance, normal recessiveness, but it's a kind of Punnett square that allows you to predict the offspring that 2 organisms will have, but it looks at 2 characteristics at a time, not just one. Our previous Punnett squares have looked at a single trait, like color, for example. Dihybrid crosses look at multiple traits, 2 at once. For example, what we're going to use are flowers. The 2 traits we'll use are color and height. Red is dominant over white for color. Tall height is dominant over shortness. Okay. So, actually I drew these guys incorrectly. Let me fix that real quick. So both of these flowers are going to be hybrids, meaning they will be homozygous for both traits. And since they're homozygous, it means they've got a dominant allele for both color and height. Having red flowers and being tall are both dominant. So that's why I had to redraw these guys. They both have to be red and tall. Okay. Here are their genotypes. This is called a dihybrid cross because hybrids are heterozygotes. And these guys are heterozygous for both characters. So how do you set this up? Well, the first thing is you've got to figure out what possible sperm and egg each of these individuals could produce. So remember, each sperm cell or each egg cell is only going to receive 1 allele for each trait. Okay, so we've got to split these up. Okay. But each egg is also going to receive 1 of each gene, 1 allele for each gene. So while we can only have a single color allele in each of our eggs, for example, this egg, if it received the dominant R for color, that's the only allele it can have. It also gets, though, 1 of these alleles for height. Maybe it gets the capital T allele for tallness. Okay. Since this happens randomly, which of these alleles is placed in each egg versus which of these ones is placed in each egg, any possible combination of these letters is possible. So you could get the capital R, so dominant R, lowercase, dominant T, lowercase. You could get this capital R partnered with the capital T or the lowercase t. We've already got one of those represented. Let's write out the next one. Likewise, your lowercase r could be partnered with either the capital T or the lowercase t. Little r, capital T, little r, lowercase t. These are the 4 possible combinations of alleles you can get. Okay. It's going to be the same for this plant as well. These are your 4 dihybrid sperm or egg. Okay. In other words, this is what goes, or these are what go on the edges of your Punnett square. Let me just draw an arrow. All right, so here's what that's going to look like. All right, so along one side, you are going to have these four: big R, big T, big R, little t, little r, big T, little r, little t. And then along the top of your square, you're going to have the same: big R, big T, big R, little t, little r, big T, little r, little t. Your offspring will be any possible combination of these four alleles with the four on top, meaning you have 16 possible combinations, 16 possible offspring. Filling out this kind of square is excellent practice. I would recommend stopping right now and practicing it, and then starting up the video again. Okay, let's just assume you did that. Here's how you fill in the tree, or the square. Make sure to group your letters back together. Okay, so the R's go together and the T's go together. Notice how I'm still putting dominant first and recessive second. Almost done. Last but not least, bottom corner, and there we go. Now I welcome you to do this next part on your own, sort of as a proof of concept. But if you go through and calculate the phenotypic ratio, for this particular cross, here's what you'd find. If you went through and counted up how many of these individuals represented by this square, how many of them were red flowered and tall, so the dominant versions of each trait or of each character, you would find that, for example, this individual is red and tall, this individual also red and tall, this individual also red and tall. If you went through and counted them up, you would find that there are 9 individuals, 9 possible offspring who would be red and tall. Next possible combination would be to have still red flowers, but then be short, have the recessive version of the height gene. If you went through and identified all of those individuals, like for example here, this individual red and short, this individual red and short, you'd find that there are 3 of them. Likewise, if you do the next combination, say you have white flowers and you're tall, so recessive for color, dominant for height. For example here, white and tall, or here, white and tall. Find that there are 3 of those individuals. And then your last combination, the 2 recessives, white and short. Interestingly, there's only 1. Down here, bottom corner. Homozygous recessive for both. The phenotypic ratio for a dihybrid cross is always going to be this. As long as your 2 parents are both hybrids, meaning they have one dominant and one recessive version of each allele. So this is a hybrid. As long as both parents are hybrids, this will be your phenotypic ratio for a dihybrid cross. All right, that's the big stuff I wanted to review. I hope it's helpful.